hope you're still staying well. This is my dog, Buddy. He just wanted to say hi. Okay, all right, let's get on the lecture. All right, I'm gonna close the camera. Okay, so you might be wondering who is in these pictures. I'll talk about in a few minutes. So everybody is very familiar with DNA, and we're gonna kind of dig deeper in the DNA structure and replication in this chapter. Okay, so first of all, structure of the DNA. So we know DNA is a nucleic acid, right? And we talk about this in the biomolecules chapter. So the building block of DNA is nucleotide. Nucleotide, there we go. All right, so nucleotides usually have three components. The first one is nitrogenous base. So it's right here, but they want to kind of show you all the 5 types of bases. So they're actually in these 2 boxes. So we have 2 groups of nitrogenous bases. We have pyrimidines and we have purines. So C, cytosine, thymine, and uracil, they are 3 types of pyrimidines. And we mentioned this previously. I don't know if you still remember that. So C and T, let me rephrase this. So in RNA, you have C and T. I'm sorry, not RNA, DNA. I was thinking about DNA, but I said RNA. So in DNA, you will see C and T. And in RNA, instead of T, you are going to have U. Okay? So basically, it means in RNA, U will replace T. So you will never see T in RNA, only U. All right, now the other 2 types of nitrogenous bases, they're adenine A and guanine G. So they are purines. So remember 2 groups, pyrimidines and purines. And for me, I just remember AG, and the agriculture, AG, they are purines, and the other 3 are pyrimidines. So tell me which group of bases are bigger in terms of their chemical structure. Right, purines, because you can see there are 2 rings here, right? As opposed to only 1 ring in the pyrimidines. Okay, so that's the first component, nitrogenous base. And the second one is the sugar, and specifically it's pentose. Remember, this means 5, right? So you have 5 carbon atoms in the sugar, so that's why we call it pentose. There's a difference in terms of the composition in the sugar for RNA and DNA. So if you have a DNA, while everything is in this 2' location, so if you do not have the oxygen atom, you have deoxyribose, and that's what you will find in DNA, right? That D stands for deoxyribose. And if you do have the oxygen atom at the 2' location, you have a ribose. So that's the sugar that you find in RNA. All right, so that's another difference between DNA and RNA. So they have difference in the type of pyrimidines, and they also differ in the structure in the pentose. All right, the last component is going to be the phosphate. All right, so for nucleotides that make up DNA, RNA, you only see 1 phosphate. Okay, so this is going to be the component for DNA and RNA. But of course, you may have 2 or 3 phosphate groups. Okay? And then examples are, remember, ADP. E means di, so you have 2 phosphate groups. And same thing for GDP. So ADP, we talk about this in metabolism, right? Energy. And then GDP, this is in cell communication, right? This is how the signaling pathway works, right? Using these molecules. And if you have 3 phosphate groups, you have ATP, and here you have GTP. Right? Okay, but again, in DNA, RNA, the building blocks only have a 1 phosphate group. Okay, so make sure you know these 3 components, and if I have a figure, like this figure in the test, you should be able to identify which component is which. So I may, have a number here. So I'm going to ask you, what is number 1? So that's the phosphate group. And what is number 2? That's the pentose, or you can say that's a sugar component. Now pentose is better, right? It's more specific. And then maybe let's see if you have a cytosine right here. I'm going to say, what's number 3? Then you can say, oh, that's a nitrogen space. I do not need you to differentiate the 5 different kinds of nitrogen spaces. Okay, so you just need to know in general, okay, that's a nitrogen space. All right, so DNA has a very unique structure, and we call that a double helix structure. So you can look at this picture or this picture right here. So it looks like really like a ladder, but instead of being straight and kind of two-dimensional, they form, this ladder kind of forms that kind of helical structure. So that's a typical DNA structure. I don't know if you have seen this, but I have been to Chapel Hill campus, and we kind of walked past a medical research building, and then the stairs, because you can see the wall is just glass, so you can see through. They have very fancy stair that's in the shape of a DNA helical structure, which is pretty cool. Okay, now we have to mention who discovered the structure of DNA, because this is the foundation for biology. This is probably one of the most important discoveries in the history of biology. Okay, I'm going to turn up the volume a little bit because someone's running the lawnmower, creating a little bit noise in the background. Okay, so the DNA structure was discovered in 1953 by an American and an Englishman. So their names are James Watson and Francis Crick. So you have to memorize those 2 names. They were such important figures in the discovery of DNA. So this is a very famous picture with Watson and Crick, and that's the DNA structure that they proposed. So this is one of the most famous pictures, the iconic picture in the history. All right, but their discovery was really kind of, they might not make the discovery without the help of a female scientist named Rosalind Franklin. So this is her. Because she actually discovered the X-ray diffraction pattern of DNA. And with this picture, the X-ray picture right here, Watson and Crick was able to kind of figure out the structure of DNA. So earlier, the picture that you saw was, oh, I don't want to do that. You know what, I'm going to discard. So the pictures that you saw right here, these are actually Rosalind Franklin. Yes, I was looking for her picture. And you can see that she was even on the cover page for Time. Because later people discovered that she actually contributed significantly to the discovery of DNA. But back then, that female scientists were not really given any opportunities. So it wasn't really kind of equal between males and females in the scientific world back then, a few decades ago. So that's why she wasn't recognized until very recently. All right, now we're going to look closely at the DNA structure. So remember I said DNA is like a ladder, right? So this part is going to be the phosphate sugar backbone. Okay, so this is the backbone. And it's made of the 2 components in the nucleotide, the phosphate group. Here, let me use this picture, the phosphate and then the sugar. So that's the backbone. And in the middle, I'm pretty sure we call this part of a letter run. Okay, so the runs represent the base pairing. Okay, so the 2 bases from the 2 strands, like if you have another base right here, they will be kind of bound by hydrogen bond. So this would be the run of the letter. Okay, so the backbone is linked by the phosphor diester bond. So phosphor, because you have a phosphate group right here, and then diester, di means 2. So you have 2 ester bond. So this is 1 ester, and then this is the other ester bond, right? So you can see that the backbone, this part of the backbone is connected by the phosphor diester bond. All right, okay, so remember that. Now, there are some other structural details. The first one is double-stranded helix. So DNA has 2 strands. Remember that. because that's another difference between DNA and RNA. So DNA has 2 strands. And RNA, can take a guess? RNA has only 1 strand. Okay, so that's the third difference between DNA and RNA. So the double-stranded helix has the sugar phosphate backbone. We'll talk about that. And it's always going to be on the outside. So that's going to be this part right here. Or in this picture, that's going to be the backbone. And then the base pairs are going to be the inside, right? So this is a base pair, base pair, base pair right here. So they're going to be the kind of the run of the letter. The second structure detail is that the 2 strands run in opposite direction. This is very, very important, especially when we start talking about DNA replication and DNA transcription. So they run in opposite direction. How do we know they run in opposite direction? So we have given the direction to each strand. Okay, so one strand is going to be from 5' to 3'. And I'll talk about how you determine the numbers in a second. Okay, so 5' to 3'. And then the other strand, the complementary strand, is going to run in the opposite direction, which means it's going to go from, okay, so you have to go from 5 at the bottom this time, right? And then 3 on the top. So the other strand, if you read it from top to bottom, it's going to be 3 to 5, right? So this is kind of opposite direction. 5 to 3 for one strand, and then 3 to 5 the other, for the other strand. The right-handed helix. So it's kind of hard to demonstrate this without seeing me, but let me see if there's a way to do that. You know what? Let me finish the rest of it, and then I can exit my PowerPoint. The last feature is that you are going to have a major and a minor groove in the DNA structure. So look at the bottom picture right here. You can see this is a larger groove, right? So that's the major groove. And then over here, this is a smaller groove. So this is going to be the minor one, all right? So you are going to see major and minor grooves in that double helix structure. Now these structures, or these grooves, are pretty important because these are the binding site. Some of the text are apparently covered by the picture. So these are the binding sites for DNA binding proteins during processes like replication and transcription. So when we get to replication, you will see that there are a lot of workers that work on that process to replicate the DNA, and those workers are proteins, enzymes. So those enzymes or proteins, they need to bind to DNA to do their work, right, to replicate or transcribe DNA. So where do they bind? They usually bind to these grooves, minor and major grooves. Okay, now here's the question. What keeps the 2 strands held together? Okay, so the answer is going to be here, the hydrogen bond. I don't know if you remember the hydrogen bond. Remember we talked about ionic bond, we talk about covalent bond, and the weakest one, the last one, is the hydrogen bond. So the 2 strands are held together by the hydrogen bonding between the nitrogenous bases. So you can see there are 2 hydrogen bonds, right? There are 2 bonds between A and T. And then how many for C and G? 1, 2, 3, right? So C and G will have 3 hydrogen bonds. So if you look at the 2 strands, this is base pairing in the middle, right? So what keeps the 2 bases connected? The hydrogen bond. Right? So why do we use hydrogen bond to keep the 2 DNA strands together? Because the hydrogen bond is kind of strong enough to keep the 2 strands together normally, but it's also kind of weak. So it's pretty easy for enzymes to break this hydrogen bond and separate the 2 strands when you need to replicate a DNA or transcribe DNA. In order to do those things, you have to separate the 2 strands first, and then you can work on each strand to replicate or transcribe. Okay, so basically the hydrogen bond is strong enough to usually keep the 2 strands together, but it's also kind of weak enough for the enzymes to break the bond and separate the 2 strands when needed. Okay, so that's the hydrogen bond between the 2 strands. Now, I may ask you another question. So what keeps the backbone together? I think I have a question here. What keeps the backbone together? What keeps the backbone kind of linked together? And the answer will be the phosphor diester bond. Okay? So make sure you differentiate whether I'm asking you the bond to keep the backbone together. That would be the phosphor diester bond. Okay. And I could also ask you what keeps the two strands together. So that would be number 2. And the answer would be the hydrogen bond. Okay. All right, so let me exit this, and I want to show you the right-handed helix real quick. Okay, so use your right hand and then make this shape. And make sure the thumb is pointing up. So the direction for your fingers, not thumb, the rest of the fingers, this is the direction that how the helix is gonna form, is gonna kind of curve and go up. So that's gonna be the right-handed structure. So the helix is gonna kind of curve and run this way. Okay, so that's what it means. All right. Okay, not sure if that's helpful, but hopefully, don't worry about that too much if you still don't understand. That's not the major point. So if you still have questions about that, feel free to ask me. But again, it's not going to be very important information for this chapter. Okay, so the next slide talks about how we use DNA or why we use DNA to store genetic information. So in order for DNA to function effectively at storing information, there are 2 key processes. Okay, so the first one is really just saying that in order to store genetic information, you have to be able to copy that information every time a cell divides and make minimal errors, right? Because you want to keep the genetic information consistent between the different generations. And you don't want to make too many errors because it may cause some really bad mutations, which lead to reduced survival or reproduction. So you must be able to copy that information, and then you don't make too many errors. And then this can be definitely achieved by the structure of DNA. And we'll talk about that. Okay. And the second requirement is really that that information has to be able to translated or expressed to the functional products, which are proteins. I think I've asked you guys a few times about this. So the information of a DNA has to be passed on to make what functional product, and that will be proteins. So that process of genetic information flow from DNA to proteins, that's called a gene expression. Okay, so you have to be able to use the information in DNA to make the correct proteins. So that's the 2 requirements. You don't have to memorize that, just make sure you understand. Okay, now talking about genetic information. This information is stored in DNA, but how do you store information? It's really stored in the sequence of the 4 bases. Remember when we talk about nucleotides, everything else is the same, right? The pentose sugar is the same, the phosphate group is the same. So they are pretty consistent. There's no variations in those 2 components. The only component that you can change is the nitrogenous base, right? So that gives us a way to store the information, and really lots and lots and lots of information. So think about the order. You may have A, T, C, G, right? You may have A, G, C, T. You have so many ways to do different combinations of the different nucleotides because they have a different basis. So the DNA sequence is really determined by the 3rd component, the nitrogenous base, because you have these 4 options in DNA, and then when you have millions and millions of nucleotides, you have these kind of astronomical number of combinations. So everybody will probably have unless you're identical twins, you will have a different DNA sequence than anybody else. Complementary bases form hydrogen bonds with each other within the double helix. So we already talk about this, right? So the base pairing, and that's achieved by forming hydrogen bond. And usually when you have the hydrogen bonding, you are going to have a bigger base, which is purine, paired with a smaller base. And it's always that case, right? So A pairs with T, A is purine, and T is pyrimidine. Over here, G is purine, and C is pyrimidine, right? Oops. All right. So you always have a small and a large kind of pairing up. So that keeps the width of the double helix constant. Okay, so you have pretty much kind of the same width throughout the DNA molecule. All right, now this is super important. This really has to be carved in your mind. So A pairs with T, and then C pairs with G. Okay, now I have a question for you. So let's see, where is the DNA? Ooh, I have a typo here. So it's really, where is the DNA information stored? So take a second. What's your answer? C, right? The sequence of bases. All right. Next question. If we know that DNA of a particular organism has 30% adenine, what percentage is cytosine? So again, I would recommend that you pause the lecture video and then work through this question on yourself, and then come back and we'll review the answer. Okay, so we already know that A pairs with T, right? And then C pairs with G. So if you have 30% A in the DNA, that means T should be also 30%, okay? And 30 plus 30, so in total, that's going to be 60%. So that leaves C and G in total a 40%. And we know C is the same percentage as T because C pairs with G, C pairs with G, so C and G should have the same percentage. So in total, they have 40%, and each one accounts for half of that. So 40% divided by 2, that's going to be 20%. So 20% for C and 20% for G. So cytosine is going to be 20%. Yeah, so I probably have this question on the test, and this really kind of tests your knowledge about the base pairing. So A pairs with T and C with G. All right, now this is just kind of information about, you are going to pass the information, genetic information from DNA to RNA first, and then to proteins. So you can use the information in RNA to make proteins. So you guys can read through this. I'm gonna just pass on to the next slide. Oh, there is a important piece of information here. That's why I kind of have this thing in dark, bold text. So some specialized genes contain instructions for making functional RNA molecules. Okay, so remember I talked about that we use the information in DNA to make proteins, right? But there are some genes that do not make proteins. So instead, they will provide instructions to make RNA molecules, because we need those RNA molecules to kind of make proteins. So for example, we are going to have transfer RNA, the tRNA, to kind of carry the amino acids to the ribosomes, kind of to provide the raw materials to make proteins. And we also have ribosomal RNA, which is part of the protein making machines. Okay, so not all DNA genes will make proteins. Some of them may make other molecules that are important. So examples are, tRNA or RNA. All right, now real quick, a comparison of chromosomes between prokaryotes and eukaryotes. So for prokaryotes, you don't have a nucleus, right? So instead, you just have an area where you can find the DNA, the chromosome. So that area is called nucleoid. So usually prokaryotes have a circular chromosome. And remember, chromosome is a complex of DNA and histone proteins, right? So in prokaryotes, usually you have a circular chromosome, which can be found in the nucleoid area. And then for eukaryotes, we have a separate compartment where we store the chromosomes. So all these lines you see, those are going to be chromosomes. And for eukaryotes, our chromosomes are linear. Okay, so they're more like threads instead of circular. Okay, so circular only for prokaryotes. And then in this area, a lot of people kind of tend to forget what this area is for. Now this dark area, which can be seen pretty clearly under the microscope, is called the nucleolus. So the nucleolus is where you assemble RNA. This is where you make ribosomal RNA. Okay? And then the ribosomal RNA will come out of the nucleus. You see these little dots. Those are ribosome, right? So ribosome is partly made of rRNA. Organization of eukaryotic chromosomes, we talked about this before. So just real quick, double helix DNA, that's the basic structure. And then DNA strands are very long, so they're more likely to kind of tangle up and break. So what we do is we are going to have these DNA strands wrap around histones. So histones are proteins. So they are kind of like, I think we talked about this in class, in yours, like a sewing kit. They're like a spoon, and then you can wrap the DNA around it to keep the structure kind of stable. And then you are going to, the next level structure is nucleosome. So the nucleosome is kind of like this, right? So the yellow is the histone proteins, and then the blue lines, those are DNA molecules. And then further, condenser will produce chromatins, but chromatins are like more thread-like, right? And then right before cell division in the prophase, remember, mitosis, prophase, the chromatins will condense, and that's when you see chromosomes. All right. So there's a video there. You can actually look at the video. It's a pretty cool animation to show you how DNA kind of is condensed, how it like it coils to form all these structures. So history of DNA. I just want to mention one experiment. Okay. So before, we really didn't know which molecule was the genetic material. Okay, so we know there's something that determines, how our body looks like, how our body functions, but we didn't know which one. Okay, so a lot of people thought back then that proteins carried a genetic information, but there are a few experiments that really proved that DNA is actually the genetic material. So I just want to mention one experiment. It was a really cool experiment. So here is the experiment. This experiment was done in 1952. Okay, so kind of almost right before they discovered the DNA structure. So let's see. Hershey and Chase used a very cool group of organisms to do this experiment. They used phages. So what are phages? Their full name is bacteriophages. You know what bacteria is, right? So bacteriophages are viruses. Cool, right? Now I'm sure you're very sensitive to this word because we're going through the coronavirus outbreak. So these viruses, they only infect bacteria cells. They only affect bacteria or bacteria cells. So phages are not going to be harmful to us because they don't want to infect our cells. They can only harm bacteria. So these two scientists use a specific type of phages called a T2 bacteriophage, and they kind of purposefully let T2 bacteriophage to infect E. coli. Remember E. coli is a type of a bacteria? It can kind of cause some symptoms in our digestive system. So this is their experiment. So this is the layout. They did two experiments. And then in these two experiments, they labeled the phage using isotopes. So they used phosphor 32 and sulfur 35. If you don't know what isotopes are, just do a quick Google search and get familiar with it. So these two isotopes, they're not common in nature. Okay, so they have a different number of neutrons than the more common form of phosphor and sulfur atoms. So they're not common in nature. So they have a very, very small percentage. Okay, so you don't usually see these isotopes. So these isotopes are used a lot in scientific experiments because since they're not common, we can kind of label different molecules with isotopes to kind of track these molecules. How are they kind of transported in your body? How do they break down? And when they break down, what kind of molecules they become? So this is a very good tracking system using the isotopes, because in your body, you don't have those uncommon isotopes. So once they see the isotope, they know, okay, it comes from the chemical that we purposefully labeled. So in these two experiments, they labeled the T2 bacteriophage. In experiment 1, they labeled phosphorus. Okay? And then in experiment 2, they labeled the sulfur. Okay? Now, do you know why they label these, or they use the two isotopes for those two elements? So think about that. So where can you find phosphorus, this element? What kind of a biomolecule contains phosphorus? You remember? What are the three components of nucleic acid? Nitrogenous base, sugar, and phosphate, right? So DNA will have phosphorus. All right. Now, how about proteins? We talked about the structure of proteins, and I'm pretty sure we mentioned some of the functional groups in proteins that contain sulfur. Okay. So proteins. Okay. All right. Now, before the experiment, that we already know that when the bacteriophage infects a bacteria cell, they're going to inject their genetic material into the bacteria. So we use the isotope for phosphorus and the sulfur, and then we are going to see which one the bacteriophage will inject into the bacteria. So we're going to kind of look for these isotopes in bacteria. If we see phosphorus 32, that means the bacteriophage injects DNA into the bacteria cell. So DNA is the genetic material. If we find sulfur 35 in the bacteria cell, then we know, okay, the virus injects proteins into the bacteria cell. So proteins will be the genetic material. And unsurprisingly, they found phosphor 32 and infected the bacteria cells. So this is how we know that DNA is actually the genetic material. Okay. Of course, there are some other experiments that also proved that DNA is the genetic material. So once we look at all the experiments, the results, the conclusion is pretty clear. Okay. I want to use this example. It's also because that we have been kind of developing antibiotics. You guys know that we use a lot of antibiotics to treat bacterial infection, right? But a lot of these bacteria have developed resistance to antibiotics. So we're developing new ways to deal with that. And one thing that scientists have been working on is to use phages to treat bacterial infection, because phages they infect the bacteria cells, right? So the bacteria don't usually develop, the resistance to those viruses. And also it's pretty safe for humans because they don't really harm human cells. So that's just an interesting kind of fact. Okay. All right, I think I'm going to stop here for first part of the video so you guys can take a little break.